Number one is coliform count of water sample by MPN test. The first one is coliform count of water by MPN test. Second one is Amylase test, and another one is nitrate reduction test. Now out of that, the first one is coliform count of water sample by MPN test. Now what is the objective of this practical? See, if you will learn to this practical, you will be able to find out whether the water is potable or it is non-potable. And to find out whether the water is potable or non-potable, coliform bacteria are used as indicators. Coliform bacteria are those bacteria which are seen in the colon of human beings and animals. They are used as indicators for water pollution. Now, the quality of water, the potability of water will be determined by using three different tests together. That three tests are: number one, presumptive test; second one is confirmed test; and third one is completed test. Now here, out of these three tests, in your syllabus, the presumptive test is included. Confirmed test and completed test is not included in the syllabus. Now from this presumptive test or MPN, you will get the idea about potability of water, or you will get the idea about presence of coliforms in water. Now to do this test, presumptive test, lactose fermentation broth is the medium used, of which the composition I have told, and you have already in the autoclave for sterilization. After autoclaving, the sterile lactose fermentation broth के test tubes you will get. Using that test tubes, you will perform MPN, that is the presumptive test. Now this test is based on statistical sampling theory. Water collected from one source at different time intervals supposed to be containing similar type of organisms in it. Practical has been based. Here, requirements for this practical are: number one, lactose fermentation broth test tubes, sterile lactose fermentation broth test tubes, single strength and double strength, water samples W1, please write in a bracket tap water, W2, sterile pipettes of 1 ml and 10 ml capacity. Procedure for this test, MPN, is sterile lactose fermentation broth test tubes, double strength and single strength, are arranged in a rows, 3 rows. Double strength lactose fermentation broth, 3 test tube in one row. Single strength lactose fermentation broth, 3 test tubes in second row. Single strength lactose fermentation. Now, after arrangement of these lactose broth test tubes in a rows, using sterile pipette, add 10 ml of water sample, each water sample, to all all 3 test tubes from the first row, all 3 double strength lactose fermentation broth test tubes into all 3 test tubes, preferably single strength, from second row. .1 ml of water sample, 0.1 ml of water sample is added in a quantity, 0.1 ml of water sample is added to the third row, which is containing single strength lactose fermentation broth medium, sterile single strength lactose fermentation broth medium. After addition of all the test, after addition of water to all the test tubes, incubate all the test tubes at 37 degrees Celsius temperature for 24 to 48 hours. After incubation, after 48 hours incubation, to all these test tubes add blue litmus paper का smaller strips of blue litmus paper. See here to the test tubes, when you will add the blue litmus paper, when blue litmus paper will turn red, what it indicates? Please answer. Acid production is there. You have added the rice tube, which are added in the inverted position, and after that you have sterilized nutrient, that lactose fermentation broth. That tubes are without bubble. So if during incubation, after addition of water, if the bubbles are observed in the test tubes, what conclusion you will draw? Gas is produced. When the red color is developed, it indicates acid production, and when in an inverted Derives tube, gas bubble is entered, you will get the indication about gas production, right? Here, the number of test tubes, you have done this experiment in 3 rows. Per row, 3 test tubes, right? Now, the number of test tubes from first row showing acid and gas production are reported. In the similar way, number of test tubes from second row and third row showing acid and gas production are reported in a tabular form. The tabular form will be made like this. First column 10 ml ka, second column 1 ml ka, third column 0.1 ml ka, and final fourth column is MPN. Now, serial number or W1, that is the one more column which should be before 10 ml ka column. First column is serial number or the water type, the 10 ml, 1 ml, 0.1 ml and MPN. The observation table will be with five columns. Here serial number, that is W1, is a tap water. Now for that 10 ml of water sample may, how many test tubes are showing acid and gas production? That will be number of test tubes out of 3 showing acid and gas production. The value of corresponding MPN you will get from McCartney's table. That McCartney's table is available in one reference book. The name of book is Microbiological Methods by Collins and Lake, page number 166. title of that chapter, which is included in a book, is counting methods. Under counting methods, one of the subheading is MPN, and under this MPN, on page number 166, they have given McCartney's table for the MPN experiment, which has been performed using 3 test tubes of double strength lactose fermentation broth, 3 test tubes of single strength lactose fermentation broth, and 3 test tubes of another single strength lactose fermentation broth, 3 rows. The water is non-potable. If the MPN is 0, then water is potable. To detect, in today's practical, you are going to use 2 different types of waters. This is the W2, which you are going to use. One group will use W2, another group will use W1. Now first group will use W1, second group will use W2. Have you understood MPN ka practical?
Yes.
See, in addition, see, just listen. To other batches, I have explained the remaining test also. To for your batch also, I will explain that test. See, this is the presumptive test, where you will get idea about the presumptive evidence of coliforms. Now, if in this test you will get the results ki water is non-potable, or you will get the idea about presumptive evidence of coliforms, then the examination will be continued by doing confirmed test and completed test. Confirmed test will be carried out using BGLB medium. Confirmed test will be carried out by using BGLB medium or EMB agar medium. Or this BGLB medium about presence of coliforms. In case of EMB agar, when small translucent metallic shining wala colonies are seen, it indicates coliforms are confirmed. This confirmed test will be carried out using BGLB medium and eosin methylene blue agar, EMB agar medium. The exam using these medias, the test will be performed, and the results of the test you will get after 48 hours of incubation. Now, please try to pay attention. See, presumptive test you will get another test that is called confirmed test. Now, in completed test, what is done? In completed test, the test tubes, BGLB test tubes or EMB agar plate पे the colonies are formed. Well isolated colonies are picked up, suspensions of that are prepared, and using that suspension, the completed test will be carried out using krypton broth, and another one, that is lactose fermentation broth, will be will show presence of indole on addition of coaxial agent, and the lactose broth will again show acid and gas production. after 48 hours incubation. Or alternative method is this lactose block is incubated at 42 to 40. Now here in case of amylase test, amylase test is the test which is used to detect amylase activity, to study the amylase activity of different bacteria. This amylase test has been used. Now what is done in this test? Here amylase test requirements for amylase test: 24 to 48 hours old culture of Bacillus cereus, Staphylococcus aureus and Escherichia coli. 24 to 48 hours old culture of Bacillus cereus, Staphylococcus aureus and Escherichia coli. Second requirement is sterile starch agar petri plate. Sterile starch agar petri plate. Then third requirement is dilute iodine solution. Dilute iodine solution. Now I will try to explain you the principle of this amylase test, and then I will tell you the procedure for amylase test. Now when you will take into consideration the principle of amylase test, this amylase test, the principle, what is it? Just pay attention. In second year, semester 3, under pharmaceutical microbiology, these biochemical tests are included. What is the objective of these biochemical tests? These biochemical tests are useful for classification, identification of bacteria. Now, when you will take into consideration specific families, and that biochemical activities are those activities which are shown by the bacteria because of extracellular enzymes or because of intracellular enzymes which are produced in the bacterial cells. Amylase is the example of extracellular enzyme. Now this amylase you have studied in a previous year, that is first year semester 2. One subject, name of the subject is biochemistry. In biochemistry, in the lab, that is biochemistry practical, you have studied qualitative analysis of carbohydrates. In that qualitative analysis of carbohydrates, there are 8 to 9 different tests. Among these 8 to 9 tests, second test is called iodine test, where you test the sample solution which is given to you with the iodine reagent, and after addition of iodine reagent, dilute iodine solution, when the sample solution will give blue coloration, you will come to the conclusion that polysaccharide, that starch might be present. Same principle is used here in case of amylase test. Now here what has been taking place A plus B दोनों रहता है. So on base of the Petri plate through which starch agar is poured and it is solidified, one vertical line and one horizontal line is drawn. At one corner of the plate, base पे, right, the name of bacterium which you are going to inoculate. You will be provided with how many plates? 3 Petri plates. Starch agar के 3 Petri plates will be given to you. So on each plate write the label as BS, then on second plate write the label as SA, and on third plate write the label as EC. That SA, BS and EC stands for Bacillus ceteris, Staphylococcus aureus and Escherichia coli. In addition, on the same plate, what you have to do? You have to write roll number of one student from your group and name of the test will be common to all three petri plates. The name of bacterium will be changed on remaining two petri plates. Is that clear? In this way, do the labeling of the petri plate. Now, using sterile nitromyr loop and aseptic techniques, take a loopful of suspension and inoculate that loopful of suspension in smaller zigzag line, in the form of small zigzag line along the vertical line. Take another loopful of suspension aseptically and inoculate along the horizontal line in zigzag manner. Same procedure will be followed for remaining two organisms. Same procedure will be followed for remaining two organisms. Important instruction here: when you are doing this practical of amylase test, try to do the inoculations first using 24 to 48 hours E. coli culture, then do Staphylococcus aureus and lastly Bacillus ceteris. Incubate all the plates at 37 degrees Celsius temperature for 24 to 48 hours. After 48 hours, when you will come to the laboratory, add drop of dilute iodine solution in these four quadrants and iodine will be spread all over the plate. Here, in that area where the starch is not hydrolyzed, you will get blue coloration. Surrounding to this inoculation, if the bacteria is amylase positive, you will get colorless zone, and that colorless zone will be seen in this way. This is amylase positive. Here, if the to whom you will call amylase positive, surrounding to this inoculated area, when the colorless zone is seen, it indicates amylase positive. If the bacteria are amylase negative, then the blue coloration will be seen up to this position. This portion will be dark yellow in color. Have you understood amylase test? Another test which you are going to do today is nitrate reduction test or nitratase activity. Now what is nitrate reduction test or nitratase activity? There are few bacteria who are having the ability to produce nitratase enzyme. Now what that nitratase enzyme will do? Nitratase enzyme or nitrate reductase enzyme will convert nitrate to nitrite. Nitrate reductase enzyme will convert nitrate to nitrite, and this conversion of nitrate to nitrite will be observed by using the indicators. That indicators are sulfonalic acid and naphthylamide. That indicators are sulfonalic acid and naphthylamide. Have you understood the principle of the nitrate reduction test? Now, objective of the nitrate reduction test I already told. For classification and identification of the bacteria, this nitrate reduction test is carried out. It is because of nitrate reductase enzyme. Now, what are the requirements? Requirements are 24 to 48 hours old culture of Staphylococcus aureus, Escherichia coli and Pseudomonas aeruginosa. Now, among these three bacteria, Pseudomonas aeruginosa is nitrate reductase positive. Staphylococcus aureus will also show little bit weakly positive test. E. coli will show compulsorily negative test. Now, procedure: nitrate peptone broth, sterile nitrate peptone broth test tubes are aseptically inoculated with 24 to 48 hours old cultures of the given bacteria, followed by incubation at 37 degrees Celsius temperature for 48 hours. After incubation, to these nitrate broth test tubes add 0.5 ml of sulfonalic acid solution and 0.5 ml of naphthylamine solution. Development of pink or red color indicate the test is positive. Development of pink or red color indicate the test is positive. Out of three cultures, Pseudomonas aeruginosa and Staphylococcus aureus shows positive test, and E. coli will show negative test. Amylase test, I have told positive or negative. Amylase test में लिखो, amylase test में Bacillus subtilis is amylase positive. Staphylococcus aureus is also weakly positive, and E. coli is amylase negative. E. coli is amylase negative. Okay.
तसं नाही
Hey, just come here. 2 मिनट में मैं करता था वो रिकॉर्ड You have inoculated half part only.
Yes, sir, but it is not possible for me to just
Okay, ठीक है चलो. Just reset it. Till that time you put litmus paper in that. Litmus paper है ना, आप MPN का ही प्रैक्टिकल किया है ना आपने. See what you have done. You have streaked half portion. You have streaked this half portion, and in the second loophole you have done like this. Now how to do horizontal inoculation? Horizontal line and vertical line. That I will try to show you. Just wait. और वे 3/4 इनोकुलेट करने का, देन टर्न इट एंड रिमेनिंग 1/4 इनोकुलेट कर. See, just listen carefully. You have performed which practical? MPN, right? Na? You have performed another practical, that is amylase test. Then catalase test. Then oxidase test and nitrate reduction test. Now here what will be done? Just listen. There are two types of waters. What I will do, I will fold this paper like this. यहाँ पे there are two types of water. That waters are W1 and W2. Now you have to write combined result. W1 is 10 ml का 3 test tubes, 1 ml का 3 test tubes and 0.1 ml का 3 test tubes. और आपको मैंने I told you to xerox that MPN का table from the book. In the similar way, 10 ml, 1 ml and 0.1 ml or corresponding MPN. Now here the number of test tubes. ये क्या है? Which one? Have you added the litmus paper to that? हाँ, तो मुझे एक बात बताइए. Number of test tubes from first row showing acid and gas production. 10 ml का उठाइए. 10 ml के 3 test tubes. नहीं, hold that test tube in hand. Remove that. Just. That is also 10 ml. This one is 10 ml. Have you done addition of? See the number of test tubes showing acid and gas production. Come on this side. How many test tubes are showing the gas production, acid production? 1. That means this result is 2. Next. This is tap water, ना?
Yes sir. W1.
दूसरा टेस्ट देखिए.
1 ml.
1 ml.
W2 sir. W2 चा काढायचं ना 10 ml.
अरे हे अरेंज करायला पाहिजे होतं ना. I have not taken that one. I have also installed it.
टेस्ट ट्यूब स्टँड घेऊन ये ना. स्टँड लेकर आओ.
This is what?
W2.
W2. इसमें देखो the number of test tubes showing acid and gas production. All three are the showing na. 1, 2 and 3. Right here 3. एक मिनट.
दो लेकर आओ.
This simple common sense, she has brought the test tube bag in which not a single test tube is done. What practical you do, I don't understand. फर्स्ट ईयर में आपने प्रैक्टिकल किया है, सेकेंड ईयर में आप प्रैक्टिकल कर रहे हैं 6 मंथ्स. नॉर्मल है, टेस्ट ट्यूब देख के समझता है. इट विल बी अकोमोडेटेड इन दैट और It is what W1, W2.
W1.
You only tell me the number of tubes showing acid and gas production. I think only one. This is what? One. तीसरा. This is what? हाँ देखो number of tubes showing acid and gas production. How many? Two. Point one. ये देखो. Two. How many? Bring that journal. See MPN here. 212. 212. Where is this 212? This one.
Yes. At 92.1.
It is 211. This one is 222. This one is 22.
So 90 मतलब 212, 27.
212. हाँ. It is 27. लिखो आप. 27. And this one 322. यहाँ पर. Both waters are non-potable. Both waters are non-potable. Please tell me your numbers. Present numbers बोलो. 67,
68.
68,
70.
70,
72,
72,
73,
73, 74, 77 और
71,
71 और
86,
86, 83 और नाइट्रेट रिडक्शन टेस्ट में सल्फोनिक एसिड एंड नैप्थिलामाइड ऐड करो और वो रिजल्ट्स के बाद आई विल राइट द मार्क्स फॉर यू यहाँ पे. इज दैट क्लियर? नाउ दिस एडिशन आई विल नॉट शो यू, आपको ही करना है. ठीक है.